It's a pleasure to be here and good evening to everyone. I am going to be talking today about the role of real world evidence and observational studies, and I'm going to try to focus on oncology. I have an example that I'm going to share with you all. Let me start off by sharing my screen. Hopefully you all can see my screen.
Yes.
Okay. Before getting into my presentation, just a couple of quick disclaimers. This is intended for healthcare professionals. The second is that the opinions and the thoughts that I'm going to share with you today are mine and don't necessarily represent that of Pfizer. And it's a non-promotional meeting that we have today. So my presentation has 4 key sections. The first one is just level setting on what is real world data. The second one is what is real world evidence. Then the different types of study designs that you can use to generate real world evidence. And then I'm going to end with an example of a study that's been done using a real world data set to generate real world evidence. And I'm going to focus on prostate cancer for my particular example. Before getting into the details, I just wanted to level set in on the fact that real world evidence and real world data is gaining a lot of prominence. And there are several macro trends that are actually influencing the rationale as to why real world evidence and real world data are getting these prominence. They're very relevant, they're very timely, especially given the scenario that we are faced right now with COVID and a lot of people actually working from home. And staying at home, even things such as doctors' visits are being held virtually. And so now is the time for real world data and real world evidence to be leveraged at its maximum. In terms of the macro trends that you're seeing, the first one is about the robustness of how real world evidence has been generated. So for example, outside of India, if you look at regulatory agencies such as the FDA or the European Medicines Agency, there's an acceptance of real world evidence and real world data to actually approve products based off of it. And that's because the way we generate this data is getting more and more robust. The second rationale being that healthcare expenditures are going up significantly. We, sometimes face an aging patient population, and so the costs in general are going up. So if you want to have a sustainable mechanism to control growing healthcare expenditures, then one way to do it is to come up with different reimbursement schemes. So for example, outcomes-based contracting is very popular. And the way you can enable some of these schemes is actually through collecting real world data and real world evidence to inform it. The third is the nature of data that we are collecting itself has evolved. So for example, in oncology, you have emerging databases such as electronic health records. And when you look at the depth of data that's actually captured, you can have everything from how a patient feels, how is a patient progressing in terms of their, survival trends, the scans that have been done. All of these are being collected. So the actual depth of therapeutic area specific data is growing significantly. And that actually allows you to do research that is very critical in informing how patients are being treated. Not only is the depth of the data growing, but the way and the types of data that are available are growing significantly. So genomic data on mutations that a cancer patient might have is growing significantly. When patients report on quality of life, that is being captured in certain research areas. And so the breadth of the data, along with the depth of data, is growing significantly. And then finally, technology has really enabled us to do things that we could never do before. So when you collect a large part of this data, you have the ability to de-identify and to link these data sources. And then on top of it, use algorithms or predictive analytics to inform and interrogate these big databases. So all of these macro trends have has led to the increase of prominence of real world evidence and real world data. So now let's look at some of the definitions. So when a lot of people talk about real world data, but how is it actually defined in a healthcare space? There are 2 key aspects to this. The first one is that real world data are data that are relating to patient health status or how healthcare is is delivered to the patient. So that's the first aspect, right? So it's about healthcare status, healthcare data that's being collected. And so that's the context. When I say real world data, that's what I mean. The second aspect of it is it's data that's routinely collected from a variety of sources. And the word to focus on here is routinely. So when you're within a clinical trial, it's all protocol driven. You're expected to come in at a certain time, labs are drawn at a certain time, but that's not how the real world works. Physician visits might not always coordinate and coincide with a given protocol. So it's any data that's collected from a variety of sources in a routine manner. So that's the definition of real world data. There are several sources from which real world data can be used to inform research. I think everyone's familiar with electronic health records. This is when, a physician can enter data in an electronic database that they might have while seeing a patient or afterwards. The other source of data, which is pretty popular, is claims and billing activities. So these are data that are collected explicitly for the purposes of reimbursement. So this is data on the cost maybe of an intervention or what is prescribed to a patient. So very detailed prescription data and so on. And then you have other things such as patient registries or disease registries. These often focus on one particular disease area. And patient generated data could range from a variety of things. It could be a simple thing such as a COVID test result that you might enter in your smartphone, or it could be a quality of life questionnaire that you might fill out, and so on. So these are the different sources of real world data. What are some of the advantages of real world data, and why is it important to collect it? When you run a randomized control trial or an RCT, they can be pretty expensive. And so cost is a major factor in why real world data is much more cost effective than randomized control trials. Naturally, the amount of time that it takes to generate real world data is much quicker and faster. A clinical trial could perhaps run for years, especially in the earlier settings of cancer. And so when you want to, generate real-time data, then it's real-world data that you'll rely on. It's much more quicker. Third is representativeness. So when clinical trials are conducted to test the efficacy of a product, the patients are selected very carefully in these trials. And so they're not always generalizable and don't necessarily represent who the drug is given to in a setting. And so representativeness is an advantage for real world data. Behavioral elements such as adherence, persistence, those are certainly not collected within the context of a trial, or if they're collected, they are biased because of protocol mandated collection or consumption of product. So those behavioral elements can be easily collected. You can do comparative effectiveness. So comparing one product versus the other is sometimes difficult to do because regulatory requirements dictate how a particular trial is designed. And the example that I'm going to share today is an example of a real world evidence that's generated using comparative effectiveness type of methods. And then finally, economic impact. So trials don't necessarily collect information on cost or certain outcomes. All of that can be collected in a real world setting using real world evidence. I've talked upon the different sources for real world evidence and what are the data types that are collected. This is just a visual representation of the specifics of data that can be collected. I'll maybe touch on just one quick example. Insurance data is collected explicitly for billing purposes. And so if you were to run a study using insurance data, the components that you won't have is how was the patient feeling at the time? What was the blood pressure of the patient? What were the scan results of the patients? You might have information on what was actually administered, but you won't have full information on what was prescribed or the other way around. And so each data set has its own advantages and disadvantages. Why am I sharing this? I'm sharing this because the type of study that you do and the type of database that you actually use to conduct that observational research tells you, it tells you which data set you should be using. And it also influences what you can generalize in terms of the observations that you have coming out of that research. Now, all of this is fine, right? We've talked about the different sources of data. But really, what is important is that you have to link these databases in order to get a complete picture of the patient journey. Just one component by itself doesn't give you the complete picture. But this is easier said than done. If you have to link data coming from multiple different sources, it's not an easy task, and there are several key challenges. The first one is around linkage. I think it's very important that you are able to link and connect one patient and follow their journey. And because it comes from different data sources, what is captured in one data source doesn't always allow you to collate and link these data sets. The second is around de-identification. So, making sure that patient privacy is protected when you conduct this research is of critical importance. Obviously there are regulations and rules that govern these. You've all, I'm sure, heard about HIPAA regulations, but then, you have the GDPR regulations. So these are extremely strict regulations. So you have to be able to de-identify and comply with these, and it's not always easy to do that. The third challenge is around primary data collection. So sometimes you have data sets, but the data that's contained is incomplete. As a person who does this, on a day-to-day basis, looking at the different databases, conducting real-world evidence, one of the most frustrating pieces is that you end up with incomplete data. A patient is only followed up when they come to a particular clinic, but let's say they go to another hospital or they move cities, then you lose that link. And so you don't really know what happens to that patient. And therefore, primary data collection can be a challenge. And then finally, harmonization and standardization, with different sources and so on. Now that we've looked at real world data, and in preparation for this particular talk, I was very curious to see, what are some of the initiatives and progress that has been done in our country on building this digital health infrastructure. And I was actually very excited to identify this particular initiative. It's an initiative that's sponsored by the National Health Data Mission, and it's called as Ayushman Bharat Digital Mission. The vision is what you see on the screen here. So Ayushman Bharat Digital Mission aims to develop the backbone that's necessary for supporting the integration of the digital health infrastructure in the country. So it's a very ambitious vision. It's a very timely vision, and it's a vision that will actually really progress the conduct of observational research in India. So it's very exciting. Now, I know what some of you might be thinking, that it's going to take a little bit of time to get there. And of course, merging and doing this at a national level is never easy, but there are going to be several advantages of doing something like this. Now, imagine a patient actually entering this data, whether it's through their digital phone, because this initiative is supposed to link the different sources of data and provide that infrastructure for linkage. So it's not about developing a registry, but establishing the links. And think about the advantages from a patient perspective. If I were a patient and I was entering my data, and I now have access to the expertise of oncologists who are treating the best experts, even though someone is sitting in a village, in a rural part of India, that's the promise of actually having this sort of an infrastructure. Imagine the ease with which someone sitting in a remote area can actually share their patient records. They can get an opinion and a protocol developed from an expert doctor that's perhaps sitting in a hospital in Mumbai, like the Tata Memorial Hospital or other centers and so on. So this vision and this initiative is really, really very exciting, and I'm looking forward to how this progresses. So this is a little bit about real world data and where we are in terms of advances there, and especially what's happening in India with regards to real world data. Let's switch gears to real world evidence. So real world evidence is evidence regarding the usage and benefits and risks of a medicinal product that's derived from the analysis of real world data. So when you analyze a lot of this real world data, what you end up is real world evidence. And so it's important to differentiate the two. A lot of people use the terms interchangeably, but they have very specific and different meanings. Real world evidence can be generated using multiple different designs. and they're not limited to just trials, because a different type of trial can actually also give you real world evidence, and I'll talk about some of those trials such as pragmatic trials. But then obviously real world evidence can be generated through observational studies. And the final point to make here is, although we think of real world evidence as clinical evidence, it also includes economic evidence. So it could be the economic evidence or healthcare resource utilization, or it could be some patient relevant evidence that's also generated. The type of evidence that's generated during the different aspects of understanding a medicine is very different. And I just wanted to highlight that across the different stages of a product's life cycle. So a product goes through a development phase. It then goes, once it's launched, goes through a growth phase. And then there's the mature phase. And what we mean by that is, during development, it's the research and development of a particular product. So what do we do in terms of real world evidence during that point? It's really about understanding how is a disease state actually treated, how is it being treated in the outside world, understanding what is the standard of care, what are some of the treatment patterns, what are the products that are being prescribed, and so on. So the type of real world evidence during the research and development phase is very different from what you might do when a product is actually available. When a product is available on the market, you will focus on other aspects, such as how do patients actually adhere to it? What are some of the long-term clinical outcomes? Because a trial could only be of a short duration. So what are some of the long-term outcomes? What are some of the adverse events that you're seeing in a real world setting? Those are the type of observational research that you can conduct when a product is available on the market after it has been approved. And then once it goes off patent or some other competitor comes into the market, you can think about the effect of switching to another product. What does that do to a patient and their outcomes? How do you differentiate one product versus the other? So it depends on what you're trying to do with real world evidence, and at what phase of a product or a disease are you trying to generate real world evidence. And that varies widely. Now that we've level set on sort of the definitions, let's look at the difference between a randomized control trial and real world evidence studies that we are going to look at. In a randomized control trial, basically what you're measuring is efficacy. Is a product safe? Is a product efficacious in a highly controlled setting? In a real world setting, you are looking at effectiveness and safety and how a product is being used in everyday utilization by a patient. So these two types of mechanisms for generating data look at two fundamentally different things. And there is a hybrid in between, and I'll talk about it in the next slide. The second is study design. Obviously, randomized control trials are prospective in nature. That means you follow a patient forward in time. They're interventional, that is, a medicine is actually administered or some sort of an intervention is done within the context of the trial. And patients are often randomized in trials. There are some exceptions. In a real world setting, you could have prospective, but because data is already collected, you could have retrospective elements to it. So you're looking back in time to make some deductions or some interpretations about how a patient is treated. Most often, these observational studies may have a control group, but not always. And they are certainly not always randomized, because it is impossible to randomize, especially if you're looking back in time. A patient population in a clinical trial is very homogeneous in the sense that patients are selected very carefully. You want to make sure that it's going to be safe for them. You want to make sure they belong to perhaps a single tumor type, maybe even a mutation or a biomarker within a particular tumor type. So they're very homogeneous. They might have an ECOG score of 0 or 1, which is very specific to that particular trial. In a real world setting, you're going to have different types of patients taking a medicine. So when you conduct real world studies, it's very important to control for the underlying patient population. And I'll talk about some of these aspects when I talk about the example that I have. And then finally, outcomes. In clinical trials, your outcomes are relatively fixed. You're looking in cancer in terms of response rates, you're looking at progression-free survival or event-free survival, and you're basically going to look at safety and efficacy. In a real world setting, using a real world evidence type of analysis, you can look at other measures like persistence, adherence. You could look at patient specific measures. These are sometimes difficult to measure in a randomized control trial. Why is oncology a little bit different and very relevant to this area? It's because there are peculiar nuances in oncology that are not always found in other disease areas. The first one is you see a lot of orphan drugs for small indications and for rare tumor types. This is important because you are focused then on a very small subset of patients, and it's very hard to run large trials when the tumor type is very rare or it's driven by a particular subset of mutations. Therefore, in oncology, real world evidence is important because when you have a large amount of real world evidence that's available, you can extrapolate these findings and have a better understanding of these rare tumor types and therefore inform how patients can be treated. The second is that patients often have very poor prognosis in certain cancers, especially stage 4, or if it's completely metastasized in a patient, then it's very difficult and their prognosis is poor. And so having a randomized control trial is very difficult, and it's not ethical sometimes to randomize to no treatment when you know that there is a treatment that can be used. In this particular case, you have what are called as synthetic control arms. I'll talk about it in a couple of slides. It's basically using real world evidence to do comparative analysis with data that's observed in a clinical trial. This is also linked to single arm trials. So a lot of oncology products are approved using single arm trials. So how do you compare the efficacy from a single arm trial to what is being done in a real world setting? And very often observational studies can help you in doing these comparisons. And then finally, we've talked about outcomes. So these outcomes can look very different. And because they are done in small subset of patients, there's some uncertainty whether the product will work in the same way outside of a clinical trial in a real world setting. So real world evidence can allow you to look at this in a broad patient population. Okay, I'm going to switch gears and now get into the different types of observational research that is being done to generate real world evidence. Before we go into the different types, let's look at two key definitions that I'll refer to. The first one is exposure. So essentially, when you conduct a study, you're looking at the association between a factor and what is the outcome that's going to come because of that particular underlying factor, which could be a risk factor and so on. So that factor is referred to as exposure. In any particular study, any sort of risk factor or mutation or a predictor variable or independent variable, these are the different terms that you will hear. What this refers to is what is it that's causing the outcome and how can I identify the relationship between that factor and the outcome. The second definition is outcome. So this is really what you're trying to measure, right? What is the outcome that I'm going to measure in my particular study or my particular, real world evidence? In oncology, these are like things such as overall survival, progression-free survival, perhaps, some specific biomarkers in a patient and so on. So these are the two key definitions to keep in mind. And then the type of study that you do is impacted by how you select your patients, how do you define the starting point for your study? So when do patients actually enter the study? What are the choice of exposure variables or predictor variables that you want to identify? And this is often dictated by the database that you use. So you can only measure that's recorded in the underlying database that you have. And then finally, the choice of outcomes. Both 3 and 4 are dictated by the type of database or real world data source that you use to conduct your study. Let's look at the different types of study designs. You have 3 types of broad categories of study designs. I'm not going to touch on clinical trials because these are not considered real world data. And today I'm going to focus actually on observational study designs. There are 4 types of observational studies that I will talk about. And then there's this intersection between clinical trials and real world data. I'm not going to get into the details of these, but there are 3 types. There's an external comparator, which is basically looking at real world data to supplement clinical trials when you can't do a head-to-head clinical trial. There's pragmatic trial, and there's things such as innovative extensions. So in pragmatic trials, you do randomize, but you don't necessarily have a protocol driven assessment. It's whatever happens in the real world study and so on. And innovative extensions are just extensions to clinical trials. So that's at a high level. I won't get into the details of this, but let's look at observational study designs. There are 4 types of observational study designs, and they are all different from each other. The first one is cross-sectional. By the way, I should also point out that as you go from cross-sectional all the way to cohort study designs, the level of evidence that you get out of it actually increases, and you get a higher level of evidence when you do a cohort type of study. A cross-sectional study is a study that's done at a point in time. So at this point in time, I'm going to look at 2 cohorts of patients. I'm going to look at the exposure and the outcome in these patients. And then depending on the presence or absence of the exposure at a point in time, I'm going to assess if there is an association between the exposure and the outcome. The issue with this is you can't really establish causality. It is only an association that you can establish between the exposure and the outcomes. Case crossover is an interesting type of observational study design. In this particular case, you have patients that serve as their own control. So let's say you want to assess a very acute event that happened. Let's say you're seeing a lot of adverse events that are occurring in patients. You will look at their history before they got the medicine, perhaps, and after. And you will assess whether it was caused by that particular medicine or that particular exposure that they had. And now this is advantageous because there is no variability. It's the patient that serves as its own control in this particular type of study design. The third one is case controls. Here you start off with a group that may or may not have the outcome of interest. If they have the outcome of interest, they are classified as a case. If they don't have the outcome of interest, they are classified as a control. And you look back in time to see if they had the exposure or not. So when you do the real world assessment, you start off with the outcome and you look back in time. And that's why these arrows are facing backwards. The advantage is that you can actually look at multiple exposures in this particular type of study design, but you have to be very careful in terms of how you identify the controls. You have to make sure that they are matched with the cases and it's not variability in the underlying factors that's impacting the outcome. And then the 4th type is a cohort study. A cohort study basically follows patients prospectively. It could be retrospectively done. In fact, the example that I'm going to use is a cohort study that was done looking at patients retrospectively. So these are the 4 types of observational studies that can be study designs that can be implemented to generate real world evidence. Each of them have their own strengths and weaknesses. So for cross-sectional studies, they can be very quick, but what you can't do is establish causality. Did this exposure cause this particular outcome is not something that you can talk about. Case crossover, as I've touched on, is really good because you minimize any confounding that could be resulting from differences in the 2 groups. You know that it's the same patient that you've looked at, so you know that their demographics are being controlled for. But there's some scope for informational bias. So patients might not recall all of their exposure or exposure variability before an outcome was assessed. And so that's the disadvantage of case crossover. In case control, these are also relatively quick and inexpensive. But I've said that identifying a control group can sometimes be challenging. There might be some type of bias, such as recall bias. So you're relying on these patients or records from the past, and you might not have complete records on these. And then the association might be limited. So, how do you attribute that the outcome was definitely because of the exposure that you've seen or a drug that was given to these patients is limited. And so the best type of studies to actually do to generate real world evidence are cohort studies, because this is the one design area where you can get to causality because you're following these patients up very systematically. And so the sequence of events can be modeled out. And so causality can be established using a cohort type of study. But there are some disadvantages. You sometimes require very large studies, especially if the cancer type that you're looking at is rare, or there's a long-term follow-up that's required. And so they can be much more expensive, and you have less control over some of the variables if you do this in a retrospective manner, i.e. looking back in time. Okay. I'm going to move on in the interest of time, but we've looked at real world data, real world evidence, and then the types of observational research. I'm going to actually share an example, and this is a very interesting example in prostate cancer of an observational study that was done. This is that, remember, out of the 4 types, this is the last type that we talked about, which is a cohort study. This is a study that's looking at comparing enzalutamide or abiraterone as first-line therapy among chemo-naive metastatic castration-resistant prostate cancer patients. It was published recently in Nature, Prostate Cancer and Prostatic Disease, and Dr. Scott Tagawa was the key researcher on this, along with several others. So this example is interesting because there are 2 products that are available or were available at the time when this study was done, enzalutamide and abiraterone. Both of these are novel hormonal therapies used and approved for first-line treatment in patients with mCRPC or metastatic castration-resistant prostate cancer. They had been approved for a while, and it was very hard to do a randomized control trial. So remember, I'd said sometimes it's difficult to do a randomized control trial because if you want to do comparative effectiveness, it's difficult to power a study that's large enough when 2 products have similar mechanisms of actions. So they had both been established to show an advantage in overall survival using randomized control trials. And there were meta-analyses that were also done using these trials. 2 of those meta-analyses showed that there was better overall survival with enzalutamide. One of them was inconclusive and showed no difference. So how do you know what's happening in a real world setting, which is why this study was done? The objective of this particular study was to assess the comparative effectiveness of these 2 novel hormonal therapies, enzalutamide and abiraterone. So that was the exposure, if you will. And the outcome that was assessed was overall survival in chemo-naive patients with mCRPC. Let's look at the methods. So we've talked about advantages and disadvantages of different data sets. This study was done using a US database called as the Veterans Health Administration database. Now, why was this particular database picked? It is one of the largest integrated healthcare systems in the US. And when I talk about integrated, it means your medical records, your pharmacy records, your billing information, all of that is integrated in this particular database. So it provides you that longitudinal patient journey that I was talking about, and it allows you to follow patients through their journey. And the other reason why this particular database was picked was because prostate cancer is very common among veterans. And so if you wanted to do a large study, this was the perfect database to use for doing this study. In terms of the statistical methods, and I know you have a few upcoming sessions on statistical methods, so I'm very sure you'll talk about this. The method that we used was Kaplan-Meier analysis to estimate overall survival or OS. And we used a regression methods, so Cox proportional hazards regression model, adjusting for confounding variables or differences in characteristics for age, race, Charlson Comorbidity Index, different comorbidities that might be different in the two groups, and whatever treatments they might have received in an earlier setting. Patients were selected as long as they were adult patients, they had evidence of surgical or medical castration, and they were assigned to two different cohorts based on whether they had enzalutamide or abiraterone. And then outcome, I've talked about this. We looked at overall survival. This is what the patient demographics look like. There was very little difference in terms of age. So the two cohorts were very comparable. Patients who got abiraterone and enzalutamide, you can see that the sample size is very huge here. You almost have 2,000 patients treated with abiraterone and about 1,200 patients with enzalutamide. There were some key differences. So the number of patients with arrhythmia, congestive heart failure, and type 2 diabetes were higher in the enzalutamide group compared to abiraterone. But these differences were controlled for when we looked at the end result. And then in terms of steroid use, you saw a larger proportion of patients on abiraterone taking steroids. This is again very clinically justifiable because these patients are recommended concomitant therapy with steroids when they are prescribed abiraterone. This is the key slide or the money slide here, right? When analysis was done using the VA data, what you saw was that there was a hazard ratio of 0.84. So what does this mean? This shows that in this large database, after you adjust it for differences in age, race, comorbidities, and treatments, you found that enzalutamide treated patients had a 16% lower risk of death versus those patients that were treated with abiraterone. There wasn't any major difference in the duration of follow-up. So in this retrospective analysis, patients on enzalutamide were followed up for 18.2 months, abiraterone patients for 19 months, so very similar here. And their time for first treatment duration was also very similar. But this is the key result. So this is an example of how you can take a large database and you're able to do comparative effectiveness, comparing one product to another, and look at outcomes such as overall survival. Now, these type of results are very relevant for physicians in informing their treatment choice along with the clinical trial results. So this is an example where real world evidence and clinical trials and meta-analysis all together can help a physician understand how two products are being utilized and what their effectiveness looks like. You might say that there might still be variability in how the results were assessed for these patients because some of them might have gone on to receive other therapies, some of them might have gone on to only being treated by one particular drug. And so it's always important to remember that when you conduct observational research, you should conduct sensitivity analysis. What you see on this slide here are sensitivity analysis results that were done in order to test the hypothesis that overall survival indeed was different. And you'll see in some cases it was consistent and in some it was not. So what do I mean by that? If you look at on the left-hand side, the subset of patients that only got first-line treatment with these agents, there was a 29% reduction in the risk of death or improvement in their overall survival when you saw patients who took enzalutamide versus abiraterone, and this was statistically significant. If you look at another subset, and that's the Kaplan-Meier in the middle, who crossed over from one NHT to another. So let's say they started on enzalutamide, switched to abiraterone, or started on abiraterone and switched to enzalutamide, you see that the overall survival is actually very comparable. And there is no difference in overall survival. Although from a trend perspective, abiraterone might look a little bit better. So patients starting on Abi switching to Enza, but there was no statistical significance. So they were very comparable. And on the right-hand side, you see a Kaplan-Meier for those patients who switched to chemotherapy. So they got first line with a novel hormonal therapy, and then they progressed on it and they were switched to chemotherapy. In these patients, it was seen that enzalutamide had a little bit higher overall survival, but here again, it was not statistically significant. You should also note that here there are smaller subsets that were picked to do these analysis. So only patients who were treated with one product, only patients who switched from one to another, and then only patients who went on to receive chemo. But all of these help you understand the overall picture. So doing sensitivity analysis in observational research is a very common mechanism for testing your main hypothesis and seeing if the findings actually hold and come through in sensitivity analysis. And then I'm going to conclude with some limitations and the end result. Limitations, I talked about why this database was good, but you also have to bear in mind that this VHA database is an administrative claims database. So things such as coding errors or an incorrect diagnosis being entered is very common in these sort of databases. Second is generalizability of the findings. You can generalize these findings to the VHA subsystem, but you have to do multiple of these studies to actually get the picture in the whole population. And then OS can be impacted by variables that might not have been captured here, such as performance status or some labs. Although these researchers did look into things such as PSA levels, alkaline phosphatase levels, which are all known to impact your result of overall survival. So in conclusion, this study showed that in this particular cohort of patients, and this was a cohort analysis, those patients who were chemo naive and were treated with enzalutamide had a longer median treatment duration versus those patients who were treated with abiraterone. And I think that was it in terms of the example that I had. I want to thank you all for attending the session. I want to thank Sanchita and Dr. Goda for giving me this opportunity to present to you all. With that,